The overall goal of this method is to prepare plant chromosomes suitable for the fluorescence in situ hybridization technique using single and high-copy DNA probes.
This method can help answer key questions in cytogenetics and genome research, such as detection of chromosomal rearrangements, ordering of genetic markers on chromosomes, and comparative analysis between different species. The main advantage of this procedure is that preparation of chromosome spreads becomes fast, easy, and efficient. Cell suspension can be stored at -20 degrees up to 2 months and used for chromosome preparation when needed.
We first had this idea of this method when we observed that the quality and number of suitable chromosomes increased dramatically on humidity days.
Germinate 10 to 20 barley seeds on 2 layers of moist filter paper in a petri dish under dark conditions for 2 days at 22 to 24 degrees Celsius. After 2 days, cut off vigorous roots with lengths of 1 to 2 centimeters from the seed by using a razor blade. Next, prepare ice-cold water by placing a 500 milliliter glass bottle containing cold tap water into crushed ice water. Aerate the ice-cold water and immerse the root tips for 20 hours to increase the frequency of metaphase cells. Transfer the roots from water to 50 milliliters of ethanol acetic acid fixative to fix them at room temperature for 2 days. After fixation, wash 10 to 20 roots with 30 milliliters of ice-cold tap water in a 50 milliliter glass beaker for 5 minutes, twice. Transfer the roots one by one.